series featuring light microscopy, which is designated a Nikon Center of Excellence. For those of you who do not know me, I am Lisa Korpieski, the Director of Communications and Marketing at the Institute for Applied Life Sciences. In today's seminar, you will be hearing from Dr. James Chambers, the Director of Light Microscopy, as well as our guest speakers, Yousef Bagheri from the U Lab in the Chemistry Department, and Joe DePietro from our sponsor at Nikon. We are hoping that with these bi-weekly seminars, you will discover what great resources that the centralized UMass core facilities offer to our campus community and the New England region. And now just a few housekeeping items. This seminar is being recorded. I recommend you set your view mode to speaker. Please stay muted during the talks. Put all of your questions in the chat and who they are addressed to, and they will be read at the end of the presentations. Thank you. Next, I would like to introduce you to Andrew Bernard, the Director of our UMass Amherst core facilities. Andrew?
Thank you. Good afternoon, everyone, and thank you for joining for our second core seminar. My name is Andrew Bernard. I'm the Director of the Centralized Core Facilities here at UMass Amherst. I'd first like to thank Lisa and Isabel for coordinating, advertising, and running today's event. And of course, thank you to Jim Chambers, Director of the Light Microscopy Facility, one of only a handful of Nikon Centers of Excellence around the world, for featuring his facility today. We are grateful for Nikon's continued partnership in keeping our capabilities second to none. The goal of these core facility seminars is to both educate our research community about the robust resources available and to hear from partners and experts who appreciate the importance of these invaluable resources. As a reminder, the UMass core facilities are open to anyone from undergrads through senior scientists, regardless of affiliation, including to outside researchers and commercial partners. From sending samples for analysis to becoming a trained user, there are ample opportunities for engagement. Additionally, there are many opportunities to fund your work in the cores. For UMass users, there are core credits available to IELTS faculty and several seed funding programs. For our external users, the Mass Innovation Voucher Program subsidizes usage for small companies based in Massachusetts up to 75%. We are here to be your partner and to help grow research capabilities at UMass. Please feel free to reach out to me directly or to any of the core facilities if you have questions or would like more information. If there are specific topics or seminars you would like us to host in the future, please let us know. As of now, we will be hosting these every other week, and we have a full docket all the way through the first week of October, and we're really excited for our upcoming content. With that, I'll pass it over to Jim, and we'll be available during the Q&A. Thank you.
Thank you, Andrew. So, like Lisa and Andrew have said, my name is Jim Chambers. I run the Light Microscopy Core Facility here at UMass Amherst. We're an open facility. We have many users from both on campus and off campus. And with the next 10 minutes or so, I just wanted to give a really brief kind of high-level overview of what we offer our users and what kind of technologies we have. So in brief, we provide very powerful resources for imaging different organisms from the biological fields, from large things to single cells to subcellular proteins and stuff like that, all the way into material science applications. We have 12 microscopes at present, and they range the full gamut of light microscopy. We are greatly indebted to Nikon for kind of sponsoring us as a Nikon Center of Excellence. So some of the things that it's a win-win situation in which we get sponsorship in the terms of intellectual sponsorship. We have beta tests for different software, even hardware. We get, and Nikon gets to use us kind of as a showroom. Anyway, it's a really good relationship that benefits both my users as well as Nikon. So just to give you kind of a rundown of some of the basic technology that we essentially have just about every form of light microscope that's available in the commercial marketplace. I have a little word cloud here of just some of the technologies we offer on our 12 microscopes. All of the microscopes can either be self-operated, as in you get trained and then you come and use it, all the way to kind of paid service. So you send your slides or your organism, and me or my staff will actually do the imaging for you. Training is a very big component of what we do. So one-on-one training is a constant thing. So another support that we offer is assistance both before, during, and after your experiments. So this comes in the form of consultations. What is the data you're trying to collect? What's the best way to get that data? Is that data actually reasonable to acquire with the instruments we have? Things like that. And then help with, obviously, during the experiments, all the way to manuscript preparation, grant submission, things like that. Image analysis is a very big focus of what we do. And Joe from Nikon will discuss a little bit of that today. So this seminar is being recorded during the pandemic for all the live people who are there. So even during the pandemic, we've been successfully training people one-on-one in a remote fashion, as well as introducing the concept of telemicroscopy, in which you can be essentially anywhere in the world and imaging your sample that's in the facility here. So we can help you design your experiments to really answer the questions that you're trying to answer for your research topic. We perform data checks once in a while. If you have questions about the quality of your data or the accuracy of what you think your data is telling you, we can help you. We can also consult with Nikon. We do this quite often. We can help develop analysis pipelines to automate both, really to automate both the acquisition side as well as the analysis side. So this can be automated imaging, maybe a low magnification to find your tissue or your sample, automatically switching to a high mag objective, collecting the data you need, and even at that point, offloading that for an automated analysis routine. One of the benefits of being the Nikon Center of Excellence is that essentially most of the microscope software is identical. So this makes it very, very easy for trainees to switch modalities. For instance, someone who starts with a scanning confocal as an entry point can pretty quickly start collecting SIM or live cell or multiphoton data, because the familiarity of the software makes it very, very easy. So just a bit about me. Well, that's a picture of me from pre-pandemic. So I have a PhD in medicinal chemistry and molecular biology. I've been in the field of chemical neurobiology or thereabouts for about 20 years. This is weird talking about myself. I've been around microscopes and built microscopes and designed things all through my scientific career. As far as the rest of the team, generally we have interns that will work with the facility, assisting with training, imaging, and analysis. And we will be hiring a technician at some point in the near future. So with that, I'm going to turn over the talk to Yousef. Maybe we'll put this slide up back at the end, but here's some contact info for me. If you're interested in using the facility, simply email me. We use Microsoft Teams for all of our communications, internal communications. So if you're ever interested in learning about upcoming webinars or training opportunities that we're offering, sign up with this bit.ly link here. Okay, I'm going to stop sharing. I'll turn it over to Yousef. Maybe.
Okay, thank you, Jim. So can you just share my slides? Okay, so can everybody see my slides?
Yeah, we can see them.
Okay, so hi everyone. My name is Yousef. I'm a PhD student in Professor Miju's lab at UMass Chemistry. So first of all, I wanted to thank Light Facility for giving me this opportunity to talk about part of the research that I'm doing at UMass using light facility here. So today I will be talking about the application of film technology in studying the membrane transient interaction using our DNA zipper. So during last few decades, this was always the question whether the cell membrane is a homogeneous distribution of different proteins on the integrated lipid substrate, or it's heterogeneous distribution based on their tendency to different places. So currently there is more and more evidence that coming up that shows that indeed the cell membrane is not just a simple homogeneous distribution of the lipids and proteins, but it is a more organized heterogeneous distribution of this biomolecule on the membrane bilayer. So now it's kind of well accepted that on the membrane bilayer, isolated domain is formed with a specific composition of lipids and proteins. So these domains are usually a nanometer size, and they are referred as a lipid rafts or lipid nanodomains. These nanodomains, depending on the cellular process happening and the condition it has, they can change their size and also their shapes. So it's pretty much a dynamic structure happening in, on the cell membrane. So more and more evidence are being found now that these nanodomains or lipid rafts play a role in cellular processes. Like, for example, one of the well-studied area is that immune signaling is highly controlled and highly dependent on these nanodomains. For example, in T-cell signaling, having these domains is quite critical for a TCR complex formation and then TCR signal activation and basically continue. Also, it's known that pathogens usually get to their receptors that they are in the RAF domain. So this is basically a connection point that they connect to their host cells. And more recently, there is evidence linking these nanodomains to several types of cancer. And actually, there is some drugs targeting these lipid domains to cure cancer. So these domains are quite important and like worth doing extensive research on them to understand how they form and how they function. So far, it's kind of known that some particular interactions drive formation of this nanodomain. For example, it turned out that interaction between sphingolipids and estrols, for example, sphingomyelin and cholesterol, is one of the driving force that triggers this interaction in this heterogeneous domain formation. On the other hand, lipid-protein interactions through the particular lipid binding proteins or based on their lipidated domains, their interaction is another factor that drives this nanodomain formation. So, however, it's not quite clear the exact role of each of these parameters, and like it's not known that which one is the dominant and how they behave under different conditions. Mainly because these interactions are very fast. So they are in the range of microsecond to millisecond, and that makes them very hard to detect based on the existing methods. So the methods I'm referring is usually the microscopy methods, because they are the methods that we can indeed see these interactions and like get a better sense of them. So that's, and another reason is that these interactions, as I mentioned, usually the size of this nanodomain is in nanometer size, which is well below the resolution of the normal microscopy methods that they have like high temporal resolution as well. So it's quite challenging to understand and study this interaction. So this question was kind of fascinating for us. So we decided to do research on this to develop a new methodology that can help us to understand this interaction better. So for this purpose, we developed this new technology called DNA. We call it DNA zipper because it works similar to a zip. So what happens here is this method helps us to actually monitor this interaction in molecular level and also in cumulative entire cell membrane level. So let's compare this method with the traditional method, like FRET method that people used to use. So in traditional method, you basically have two targets, for example, two lipids that you modified them with the donor and acceptor. Then when they interact to each other, you expect to see FRET signal. When they are off, you don't expect to see anything. But since usually the dwell time between this interaction is very fast, so you don't really see any signal, any FRET signal. And it makes it very hard to actually understand this interaction or study them because you don't really see them. But in our technology, what happens is we have a small piece of DNA between the target and also the dye that we are using. So what happens is when they interact with each other, this DNA hybridization form between the donor and acceptor, and this elongates the time that we can see FRET for slightly longer. So here, the time that I'm talking is not really long. So this DNA is quite short, like it's 5 to 10 nucleotides. So it's very short to not induce any interaction by itself. Just lock the interaction whenever it happens, so we can see the FRET between them. So with this method, we can have a longer time that these two are close together, so we can image them more effectively than before. So for imaging this interaction, we use film microscopy. So let me just give a brief background about the film. So film basically works based on the measuring the relaxation kinetic of fluorophore when it's in excited state. So if we look into the fluorescence diagram, the normal fluorescence diagram, we have our dye in ground state. We excite it with the excitation laser. We take it to the excited state. So then it will emit a light that it's going to be the fluorescence signal. So the time that the fluorophore spends in its excited state is called the lifetime. And it basically depends on the environmental condition and the molecule that it can interact with. So if we provide a secondary way for this excited state to relax, then we will end up having a different lifetime. For example, if we have an acceptor next to this donor, so what happens is the donor gets excited, it non-radioactively transfers the energy to the acceptor. So this energy transfer here makes the lifetime be lower compared to before. So by using this difference in the lifetime, we can show that donor is indeed interacting with the acceptor. So if we look into the decay curve of the lifetime, so we can see that when there is no acceptor, no FRET, we will have longer lifetime, and when there is a FRET, the lifetime will drop to the shorter range. So this is the, and when we basically put this back in as a color-coded image, this is the image that we will get. So this is not a real microscopy, like fluorescence intensity-based image. This is a film-based image, and every color represents the different lifetime. So we are interested in film for three main reasons: because it's concentration independent, so any local heterogeneity on the cell membrane doesn't influence the result we are getting. Also, this independency that allows us to go with lower concentration means that we don't need to put too much of probe on the cell membrane, so we won't change its natural behavior. Also, we can distinguish the population of the targets. So if they interact or they not interact, they will have different lifetime. So we can easily separate them to see at which area we have more of interaction and less interaction population. And also for every excitation laser we have, there is always fluctuation, which film is not being influenced with that. So that's a great point that we can rule out that parameter as well. So here is the one example of application of film to study cholesterol-cholesterol interaction on lipid monolayer and live cell membrane. So our target here is the cholesterol, which we modify with the donor and acceptor. So when they don't interact, the lifetime is longer. When they interact, the lifetime drops. So here I'm doing a titration of donor with different concentration of the acceptor. So by increasing the concentration, there is a more chance of the interaction between the donor and acceptor, meaning that lifetime would drop. So initially we have 3.2 nanosecond, which drops to 1.2 nanosecond, and this shows the lifetime decay curve, which you can see there is a nice correlation between the lifetime and also between the lifetime and the amount of acceptor that I add. And this is the mean intensity value distribution that you can see. So for initial amount, we have only one peak at 3.2, and by adding the more and more acceptor, there is more interaction, more quenching, so it moves to the shorter lifetime. And you can see actually there is a transition between different states. So you can see like the completely free signal lifetime decreases and the quenched one increases. So that's what I meant by population separation between these two. So after confirming this, we moved into the real cell experiment to see whether we can indeed monitor this dynamic on the cell membrane or not. So for this, again, I'm using cholesterol as a target. When there is no interaction, they have a longer lifetime. When they hybridize, I add the acceptor, they transiently interact with each other. They form this hybridized form, so lifetime drops. Then what I did here, I said, okay, if I add the complementary sequence to the acceptor, so it will release the donor. So it should, I should recover the film, the same lifetime as before. So basically, it's going to be longer lifetime, drop, and increase in the lifetime. Which here is the result. So at 0.0 minute, I don't have any acceptor, so it's just a donor. There is no fret, and the film is perfectly showing 3.4 nanosecond. Shortly after adding the acceptor, I can see the acceptor signal. There is fret is happening right now, and you can see over the time the film lifetime decreases, meaning that film can replicate exactly the fluorescence signal. And at this point, I add the acceptor complementary DNA. It means that the donor will be released, and you can see the donor signal is recovering, fret is decreasing, and also the film signal is recovering. So basically, we show that film can nicely show and report the dynamic interaction on the cell membrane. And also, so these data are all in 2 dimension. So right now we have some data with the new film system that we have that it can indeed get the same data in 3 dimension, which is great because we can show the differences at different region of the cell surface as well. So by that, the conclusion is that we developed the DNA zipper that we can indeed report quantitatively the transient interaction on the cell membrane. and use that to understand the fundamentals of membrane dissociation to different regions in a better form. And also film is one of the techniques that I'm using in this project, and it's nicely can report the transient interaction on the cell membrane. So by that, I'd like to thank you for listening, and I'm sorry, I think I took a little longer than what I was supposed to. Any question I'd be happy to
I think
answer.
we're going to hold questions until the end via chat, and then they'll be addressed to you, hopefully, Yousef, if people have questions for you.
Oh, I see. Yeah, sure. Okay.
You can stop
Great.
sharing, and then we can let
Sure.
Joe start. And Joe, you can just reintroduce yourself if you don't mind.
Okay. So I think I should be.
Hi, my name is Joe DePietro. I am the Senior Biosystems Application Manager for Nikon. And let me just set up my thing here. Okay, can everybody see my screen?
Yep, we can see it.
Okay, excellent. So thank you all so much for having me today. As I said, my name is Joe DePietro, and I'm the application manager for your region for Nikon. And so today I'm going to talk about Nikon's implementation of deep learning in image processing. And I'll start with a couple slides on how deep learning is done, but I'm going to focus mostly on the tools, the exciting tools that we've developed in the AI space. So just a recap, essentially deep learning and artificial intelligence utilizes these things called neural networks. It's essentially a computer representation on how the neurons in our brain are connected and how they process information. And so essentially what you have is an input layer, which is maybe the data that you're trying to process. And this data is made up of nodes. It's essentially an array or a vector. And then it is connected to other nodes in another layer. Some processing is done, and then you have an output. Now, in a case like this, this is known as a fully connected network type here, where all the nodes in one layer are connected to all the nodes in another layer. This isn't really the kind of network that we use, though, for image processing. What we use instead is something known as a convolutional neural network. Now, these kind of neural networks, they're kind of based on the way that the visual cortex works, which I guess kind of makes sense if you're trying to process images. Then what better way to do it than to try and emulate the way our brain does it? We're really good at recognizing patterns and identifying objects in a scene. And we do it so easily and so seamlessly that it almost seems like it isn't really a difficult thing to do. But when you try and program a computer to do it, you start to realize how difficult and how complex of a process this is. So the way that a convolutional neural network works is rather than every node in the input layer being connected to every node in subsequent layers in the network, essentially what you have are receptive fields. The same way that in the visual cortex, if you look at a neuron at a particular place, it is actually responding to some kind of change, whether it's dark or light or movement, within a certain part of the visual space. So it's the same kind of thing. Essentially what we're doing is we're convoluting representations in subsequent layers. And what you can do with this is you can actually train the algorithm to detect certain features and then be able to just feed it something else and it'll be able to identify those features. We have now a few different algorithms, and I'm going to go through them for you. Some of them, well, one of them in particular is already trained. So it's already been trained to do what it's meant to do. And then all you have to do as the user is provide the algorithm your data, and it'll perform the image processing. Some of the other algorithms that I'm going to talk about today actually give you the ability to program the algorithm, to train the algorithm on your specific data, and so that you can utilize it then for subsequent data sets. So let's go through a few of them. The first one that I want to talk about is called Denoise.ai. Essentially what this one does is it removes the shot noise that's inherent in confocal images. Specifically when you utilize our resonance scanner. So some of you may be aware of this if you have some more experience with imaging, but one of the things that you can control with a confocal microscope is the dwell time per pixel. And the shorter the dwell time, the more of the signal is going to be made up of noise. And so you kind of run into this dichotomy where if you want to go fast, you have a noisier image. So the resonance scanner is great. It allows you to go fast. So how can we utilize that and still pull a nice, clean, noiseless image out of that? And the answer is with Denoise. Essentially what Nikon did, is they trained this algorithm with thousands of resonance scanning images from all different versions of the A1R system, and trained it to identify the shot noise in an image. And so now you can essentially put in any resonance scanning image that you take using our system, regardless of the dwell time, and be able to remove the noise and leave the signal intact. So here's another example of that, where we're imaging fast. You can see the top image is the raw data. You can see there's a lot of noise there. And the bottom image is the same exact image with denoise tied to it. So you might be asking yourself, well, is this quantitative? Because that's what's important, right? We don't want to remove any important information from the image. And so this is just one way of looking at it. Here we're just putting a line through a part of the image and looking at intensity measurements. And you can see that the curves overlay each other quite well. The only thing that's removed is kind of the squiggliness, which represents the shot noise. So we can actually maintain relative intensity and changes in intensity across the image, but remove the shot noise. This allows you to be able to take images fast, really utilize the resonance scanner, and then still come out with a high signal to noise image. Another algorithm that we have is something called convert.ai. Essentially what this allows you to do is to be able to count and segment cells without using fluorescence. Now, as you know, it's really easy to get nice labeling of fixed cells, but a lot of times people want to image live cells. And the way that we do this without doing a lot of damage is to use something like DIC or phase. This kind of image to our brains is very obvious. We can see the contrast. We can see the cells. If you ask somebody to go in there and manually count them, they could easily do it. On the other hand, computers have a lot of difficulty with this because from the computer's perspective, it really isn't that much contrast. What Convert.ai allows you to do is be able to train the network with cells that are labeled fluorescently. So then you can do your experiment with non-labeled cells and be able to create a binary on that image as if it were a fluorescently labeled cell. So for example, you would train the network with cells that are labeled in some kind of maybe nucleus label. And you would take the phase or the DIC image. We would use these data sets to train the algorithm. You can do this directly in NIS Elements. And then when you feed it just a DIC image, it will produce a DIC image that looks like it has the nucleus labeled. And then you can use that in order to segment your cells and do automated counting and all the other types of things that having a nuclear label allow you to do without having to do any other manipulation in your cells and without having to do any without having to have any of the damage associated with doing fluorescence imaging. Another algorithm that we have is known as Enhance.ai. This one is kind of similar to Denoise in the sense that it allows you to take images fast using like a SCMOS camera, for example, where if you're using a really short frame, a really fast frame rate, you'll get a lot of noise in the background, this kind of like pattern noise that you see here. And what it'll do, this algorithm, is it will convert the image into one that has really high contrast and low noise. The important thing to note with this one, though, is this one isn't quantitative. Unlike Denoise, it's not quantitative, but it could be used, but it is useful for if you're doing things like counting and overlap and stuff like that, where you want to collect a lot of images really fast or you want to be sensitive to the sample, you can still get a usable data set, assuming that you're not actually measuring intensity. So it's a similar kind of thing. You provide it with a really high noise image and one where you took a really nice exposure time of the same exact image. We train the algorithm to be able to see what the differences and similarities are between a high noise and low noise image of the same frame. And then you can provide it with a low noise image, a high noise image, excuse me, and produce an image that looks like it has high contrast and high noise and low signal to noise. So it's pretty cool. And then of course you can use that image for segmentation. The nice thing is that you have your raw data. So it's not, so you can actually utilize something like this in order to do the segmentation and then go back and do the analysis on the raw data using the mask that you created with the Enhance.ai data. All of these things, if you're familiar with NIS Elements, all of these, once they're trained, can be incorporated into things like general analysis and jobs so that you can use it for on-the-fly sort of stuff as well. Segment.ai is the last one that I'm going to talk about today. Similar to the other ones in the sense that you have to, similar to some of the others in the sense that you have to actually manually segment some of your data sets. So for example, here we would have the images, we would manually segment them. So here we're using a, something that has a lot of processes on it, but you might want to you could do this on any kind of fluorescent image that is really hard to use traditional methods in order to actually create a binary on it. So you basically, where you have to do it manually. And so instead of having to do it manually on hundreds of samples, you could do it on maybe 10 or 15 samples, train the algorithm, and then you just have to provide it the raw data and it will segment it automatically for you. So pretty powerful stuff. And as I mentioned earlier, these custom analysis routines that have been trained specifically on your data can be incorporated into general analysis or GA3 so that you can utilize them in a custom analysis routine. And they can also be utilized in jobs so that you can do, because it's artificial intelligent trained neural network, it's really fast compared to other analysis routines. So you can actually utilize this during an experiment and use it to create an intelligent acquisition. So have the microscope or the software make a decision about what to image next based on what it finds. For example, you might want to image a sample until you have a certain number of cells. You could utilize a trained neural network to continuously apply this, count the cells, and then stop imaging after a certain amount, for example. And so our goal with using artificial intelligence is to allow you to save time, to be able to use less invasive techniques and then get data that you would normally get with a more invasive technique. It's accurate, it's non-biased. You can produce really nice high signal to noise images, and hopefully there'll be more stuff in the pipeline as well. So with that, I thank you all for having me, and I'm happy to answer any questions at the end about our implementation of deep learning in image analysis.
Thank you, Yousef and Joe. Those great little vignettes of the research and some of the new offerings by Nikon. Before we go into the question and answer, I did want to just let all of the users and prospective users know that we have purchased 2 licenses for this NIS.ai, and they're available on Workstation 1 and Workstation 2 for those of the connoisseurs who are already users of the facility. So we're considering purchasing a couple more depending on some feedback. So Lisa, have you received questions?
Yes, so I have a couple questions for Yousef. The first one is, do you think the complementary DNA affects the interaction of the cholesterol? And that's from Joe.
Oh, that's actually a great question. So the complementary DNA, what it does here, it basically breaks up the hybridization between the donor and acceptor sequences. So that's how it basically eliminates the FRET signal, or like recovers the film. But so we don't know if it destroys the cholesterol interaction at that point or not, because that was like for us, like to show in the DNA is kind of necessary to report this interaction. Sure. So I think that one I don't know yet. So
yeah.
And I have another one, Yousef, for you. How do you control for non-specific interactions?
That's another great question. So actually, we studied this non-specific interactions. So what I did is I used a fully hybridized acceptor DNA with its complementary. So in that case, donor and acceptor cannot hybridize together. So when I did that, actually there was no change in the lifetime or any signal we didn't get from Fred. So based on our understanding, based on those results, is that having DNA is necessary for this interaction, and there is not much of non-specific interaction that this system reports for us. So for us, it's just very tiny amount of non-specific interactions happening there.
Great, thank you. The next question is for Joe. How many trainings is the minimum for segment AI for reliable results?
Well, you know, I'm gonna end up giving a maybe not. It's a hard question to answer because it is sample dependent, and I know that that's not a very satisfying answer, but we've been able to. I've seen samples like that for segment AI specifically, trained with 10 to 20. Of course, the more that you use, it's going to depend also on the variation between your samples. If you have a lot of different shaped structures that you're trying to detect, and they maybe exist in different variations, and some of your samples have more or less of these unique structures, then you're probably going to want to train the algorithm with more base truths in order for it to more accurately be able to detect what you want. The key, though, is that you want to do the hand segmenting as best as possible, because the algorithm doesn't really know what you want. You have to tell it. And so if you do a really good job at segmenting and you replicate that across several samples, then you'll have a much better result. I don't know if that answers your question.
Thank you.
Yeah.
Are there any other questions before we close out?
Yes, I just have a practical question for Joe.
Sure.
So when you train one of the, like a segment AI, and then use it in jobs.
Yeah.
I guess, you know, maybe you can just talk through the process of that. Like, how does it slow things down, or discuss the speed? Like, is that faster? I'm assuming it's faster than doing the typical thresholding, filtering, and all of that. But maybe you could make a few comments about the speed.
Yeah, sure. So it's definitely faster because you don't have to use a lot of the memory of the computer in order to render and continuously change these thresholds. So it's nice in that sense. A lot of times, a lot of the, I don't want to say issues, but one of the obstacles that you run into when you're doing it manually is you do have, like I said, variation between your samples, and sometimes the thresholds that you've set initially aren't perfect for every single image, and so you have to have the experiment stop and you kind of change it, you wait for it to render, and then you continue the experiment. Still great, still fast, faster than having to make the decisions on your own, but having the algorithm trained on your specific images allows it to really come to that single conclusion. There's a single conclusion for your whatever image you put in. And so there aren't really any parameters to change. It makes it a lot easier. Of course, it does use up a lot of GPU power, especially for training it, but also utilizing it. And so there is that aspect of it as well. But in the sense, from the point of view of the person who's creating the job or utilizing the job or the GA3, it's going to be very similar, except you don't have to have a user interface where you're trying to make it perfect on every individual sample. It should do a really great job, assuming that it's a well-trained algorithm.
Great, thank you.
Yeah.
I have another question for Yousef. Can you use DNA zipper for in vivo interactions?
For in vivo, so you mean, I'm assuming that it means in tissue and like in animal. So we we are actually thinking about performing that experiment. So we did like a preliminary test on organoid to see whether these probes can diffuse into the live organoid and to be able to report the interactions. Actually, it can diffuse to the organoid. So I think it can be used for reporting interactions in tissue-like structure as well. But we haven't done for measurement yet.
Okay, thank you. And I have another question for Joe. Is there any time you wouldn't want to use the Denoise AI? Seems like you would always want to use it.
Yeah. Well, there's a few caveats here. So you have to have a certain level of signal to noise in order for it to work properly. And we have some tools in Elements that allow you to assess the image ahead of time to make sure that the image is usable for Denoise.ai. So one way that you wouldn't use it is if you have a really, really low signal to noise image, it's just going to make it worse because it does need some, at least a certain amount of signal in order to work. Another thing is we have some different ways of implementing Denoise.ai. So you can actually implement it live and just collect denoised images. This does slow down the collection of images, so you can't use the resonance scanner as fast as you normally would. We have the ability to do that. However, we normally recommend that you don't. We normally recommend that you collect the raw data so that you have it, and that you apply denoise afterwards. And then for your specific image, do some tests like what I showed in my presentation, you know, looking at intensity profiles and making sure that you can, on your specific data set, trust the result. You know, we want to make sure that we understand that it's impossible to know exactly what's going on in a neural network. Even if we were to divulge all the different ways that it was built, the way in which it actually does the processing of the image is hard to know. And so what I would say is that you want to make sure that you have those raw data, and that maybe you do some experiments without denoise, and make sure that when you do utilize it, you get maybe a similar result before you then take full advantage of it and do things that you couldn't do if you didn't have it. Does that make sense? Just practice good science with it. And NIS Elements gives you the ability to have both the raw data and the denoise data, and tools to assess the quality of the transformation, if you will. Yeah.
Jim, do you have any closing comments?
I guess I would just want to, thank ISLES for hosting this seminar, and especially thank you for our guest speakers, for Yousef and all of his continued work in the facility. I've learned also from helping him, which is always great. And for Joe and Nikon for supporting the seminar and you know, giving us the information, which is great. So we'll keep it up.
Yeah, thank you.
Thank you.
And I just wanted to thank everybody for coming today, and that throughout this series, we're going to be featuring more individual core facilities with their relevant technologies. Our next seminar is scheduled for Tuesday, August 25th, and we'll be featuring the cutting-edge resources using additive manufacturing that can be found in our advanced digital design and fabrication facility. So save the date, and I hope to see you there. Goodbye, everybody.
Thank you.